and welcome to Medical Dialogues, your daily dose of health and medical news. I'm Mr. Zaman and here's what I bring to you all from the world of medicine. Phage combination therapy that can precisely target IBD-related gut bacteria without harming helpful microbes. For the first time, scientists have designed a phage combination therapy that can precisely target and suppress gut bacteria associated with inflammatory bowel diseases or IBD. Elena et al. Compared the gut microbiota compositions of 537 IBD patients to healthy controls enrolled in cohort studies in France, Israel, the US, and Germany. The team found IBD patients, despite their differences in geography, ethnicity, and diet, tend to have a group of Klebsiella pneumoniae or KP strains enriched in their gut, especially in those who are experiencing disease flare-ups. When transplanting the KP into mice, the team found that mice developed a severe intestinal inflammation and tissue damage, suggesting that these KP strains may contribute to worsening of IBD. Next, Elena and his team scanned and isolated thousands of bacteriophages from environmental samples. Bacteriophages are viruses that can target and infect bacteria. They identified some 40 phages that appear to be effective against the IBD-contributing KP strains, including strains that have already developed phage resistance. The team tested the phages in various groups as a potential cocktail treatment against IBD-contributing KP strains. In these phage combinations, each of the phages use a different receptor to enter bacteria and kills them through different mechanisms. Elena and his team discovered the most effective phage combination, which contains 5 phages in suppressing the KP strains in the test tube, as well as in mice IBD models where the phage cocktail attenuated inflammation and tissue damage. The team further tested 2 representative phages in this cocktail in a phase 1 clinical trial that involved 18 healthy volunteers. The experiment showed that the phages can survive at high levels and remain active throughout the gastrointestinal tract when taken with antacids while not impacting the surrounding microbiota. Participants had no severe treatment-related adverse events. New and easy rapid test to easily diagnose cancers and heart diseases. Imperial researchers have built a new easy-to-use test that could diagnose non-infectious diseases like heart attacks and cancers more quickly. The new test works by detecting molecular signals in the body called biomarkers, which are already used in things like COVID-19 testing, where the presence of SARS-CoV-2 genes indicates COVID-19. There are also biomarkers for non-infectious diseases. For example, prostate-specific antigen or PSA in the blood can sometimes act as a biomarker to indicate the presence of prostate cancer. The new test, called Crisprzyme, has been developed by a team of researchers led by Imperial College London, MIT, and Max Delbruck Center for Molecular Medicine in Berlin. Crisprzyme builds on CRISPR diagnostic test, which uses RNA to detect biomarkers in biological fluids like blood or urine. In their current form, these tests detect RNA and then amplify this RNA by creating many copies so that the signal is easier to read. However, these amplifying technologies must be temperature controlled to work, which requires expensive equipment. Additionally, although they tell medics when an infectious disease is present or not, they cannot provide information about how much biomarker is present, which is important for monitoring non-infectious diseases like heart diseases and cancer. Crisprzyme improves this technology by replacing the amplification process with calorimetric analysis, a method that determines the amount of biomarker present without the need for amplification. This eliminates the need for temperature control and additional steps and can also reveal how much of a biomarker is present in a sample. To eliminate the amplification step, the researchers used nanozymes, tiny synthetic materials that behave like enzymes. Their enzymatic-like activity increases the signal of the test, making the calorimetric analysis easier to read. A recent discovery of a distinct genomic characteristics of breast cancer in women with Pten, hamartoma tumor syndrome. Cleveland Clinic researchers have uncovered genomic characteristics of breast cancers associated with Pten hamartoma tumor syndrome or PHTS that differentiate them from sporadic breast cancers. The findings, published in the American Journal of Human Genetics, have important implications for the personalized management of PHTS-related breast cancers. PHTS-associated breast cancers often develop at a younger age and may progress more aggressively than their sporadic counterparts, but they are treated similarly, which underscores the need to better understand any underlying genomic differences between PHTS-associated and sporadic breast cancers. To address this need, the researchers performed exome sequencing on samples from 44 women with germline Pten mutations who developed breast cancer and compared the data to samples from women with sporadic breast cancers. They specifically investigated somatic mutations in both groups because it is posted that both copies of a tumor suppressor gene must be mutated in order for cancer to develop, which is known as Knudsen's two-hit hypothesis. Their analysis revealed that PHTS-associated breast cancers have distinct somatic mutational profiles compared to sporadic breast cancers. For example, they discovered that Pten and PIK3CA were the most frequently somatic mutated genes in PHTS-associated breast cancers, while TP53 was most frequently mutated in sporadic breast cancers. Notably, the PHTS group had a significantly higher frequency of somatic Pten mutations compared to the sporadic group, as well as a lower mutational frequency in PIK3CA, indicating that somatic mutations in Pten are the main drivers of PHTS-associated breast cancers. In addition, they found that somatic mutations in Pten and PIK3CA were mutually exclusive in the PHTS group, but not in the sporadic group. Hyaluronic acid, a naturally occurring compound which awakens stem cells to repair damaged muscle. A new study published in the journal Science reveals a unique form of cell communication that controls muscle repair. The scientists have now shown that a natural substance called hyaluronic acid, which is used in cosmetics and injections for osteoarthritis, is the key molecule that manages this fundamental interaction. When muscles get damaged, it's important for immune cells to quickly enter the tissue and remove the damage before stem cells begin repair, said Dr. Jeffrey Dilworth, senior author of the study. Our study shows that muscle stem cells are primed to start repair right away, but the immune cells maintain the stem cells in a resting state while they finish the cleanup job. After about 40 hours, once the cleanup job is finished, an internal alarm goes off in the muscle stem cells that allows them to wake up and start repair. Dr. Dilworth and his team identified hyaluronic acid as the key ingredient in this internal alarm clock that tells muscle stem cells when to wake up. When muscle damage occurs, stem cells start producing and coating themselves with hyaluronic acid. Once the coating gets thick enough, it blocks the sleep signal from the immune cells and causes the muscle stem cells to wake up. Using mouse and human tissues, Dr. Dilworth and his team also discovered how muscle stem cells control the production of hyaluronic acid using epigenetic marks on the Has2 gene. Interestingly, aging is associated with chronic inflammation, muscle weakness, and a reduced ability of muscle stem cells to wake up and repair damage, one of the authors mentioned. He further went on to say, if we could find a way to enhance hyaluronic acid production in the muscle stem cells of older people, it might help with muscle repair. The authors note that the regenerative effect of hyaluronic acid seems to depend on it being able to be produced by the muscle stem cells. That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.